So, good day, everybody. Sorry for a little bit of technical problems, but everything is set now. We can start. Today we have NOEMI ROTLLAN from Spain. She will be giving lecture on one of my favorite subjects, integrating the microRNAs and lipid metabolism, and present novel approaches how to inhibit microRNAs. I think this is one of the very hot topics currently because we are really witnessing the explosion of RNA therapeutics. So, Naomi, the floor is yours. Whenever you are ready for the next slide, just say to me, and then I will change slide, no problem.
Okay. Thank you very much, Marion, for giving me the opportunity to talk here in this Atheronet webinars. Today, which I've been told that it's more focused in the pre-doc and postdoctoral junior, and more in technological and materials methodology that is related with this inhibition of microRNAs in atherosclerosis preclinical model. So my talk, it's gonna be about a little introduction about cholesterol, atherosclerosis, and microRNAs. Then I'm gonna talk about the technical and methodologies approaches related to this. And I'm gonna focus in one example of one microRNA that it is well known to be regulating lipid metabolism and atherosclerosis, which is miR33. And also as one microRNA-based therapy that we use in this atherosclerosis model, which is the pH-low insertion peptide. Next. So cholesterol is a precursor of the steroid hormones, the bile acids, and vitamin D. And you know that is the major component of the mammalian cell membranes. And these cholesterol levels are maintained through tightly regulated and complex mechanisms that include de novo biosynthesis, the internalization of the exogenous cholesterol, and the efflux of cholesterol excess. And these mechanisms are classically regulated by transcription factors such as SRBPs and elixirs. More recently, microRNAs also has emerged as a critical regulator of gene expression, acting predominantly at the post-transcriptional level. And it is well known that the insufficient or excessive cellular cholesterol results in a wide range of pathologies, including the atherosclerosis, but also metabolic syndrome, type 2 diabetes, and Alzheimer's disease. Next. So in the serum, in humans, the cholesterol travels in the serum together with lipoproteins, typically with LDL or low-density lipoproteins that we call the bad cholesterol. These lipoproteins take the cholesterol from the endogenous or from the diet and direct it or transport this cholesterol to the peripheral tissues. And it is known that the high levels of this cholesterol in LDL particles is related with high levels of cardiovascular risk. This LDL cholesterol accumulates in the arteries and provoke that the macrophages take up these lipids and generate what we call the foam cell, and the accumulation of these foam cells is going to generate the atherosclerotic plaque. On the other hand, we have the cholesterol that is located, or it travels with HDL, what it's called the good cholesterol. And this HDL removes the excess of cholesterol from macrophages or peripheral tissue, and it's able to bring it to the liver and to the bile acids or the feces and remove this excess of cholesterol. This HDL is the only mechanism that we have to remove the excess of cholesterol, and this process we call that the reverse cholesterol transport. Next. So the atherosclerosis is the leading cause of cardiovascular disease, and almost 80 million global deaths per year, which represents almost 32% of worldwide deaths. And this, you have the risk factors that are modifiable, such as the genetic predisposition or the increase during the age or the gender. And also we have the risk factors that are modifiable, like the hyperlipidemia, the hypertension, or the diabetes mellitus. Next, yeah. And this, you can have complication of this atherosclerosis, which is gonna give you other disease related with this atherosclerosis, which are the myocardial infarction, the stroke, the also the peripheral artery disease, the angina, also the chronic kidney disease, and the aneurysm. Next. So this is the where we are going, the shape of things to come. So we have to be aware of that. Next. So typically a normal artery has 3 layers. You have the adventitia, where you have the mast cells, the vasa vasorum, and the nerves that connect with the tunica media, where we also have the smooth muscle cells, which are in a quiescent situation, and also the matrix, the matrix there, which is combined of collagen, elastin in a ordinate way, right? And on top of that, the inner layer is the intima, where the plaque formation is gonna happen, and the process or the initiation of this atherosclerosis starts with LDL particles that are in the circulation that get modified and get into the intima. This what does is the activation of the endothelial cells that they're gonna express adhesion molecules that they're gonna trap the monocytes that are in the circulation, get into the intima, where they're gonna modulate and become macrophages. And these macrophages, they're gonna be activated by these LDL particles and become macrophage foam cells. On top of that, we have the T cells that are also activated and get into the intima, as well as the smooth muscle cell. Next. So during the progression of atherosclerosis, we have these smooth muscle cell that are in the intima, that they're gonna generate these elements of the matrix like collagen and elastin. But on the same way, the T cells factors kind of like interferon gamma, what gonna do is impair the synthesis of the collagen by this smooth muscle cell. So making the fibrous cap more thinner. Also this together with, oops, sorry, okay. Together with the macrophages that are activated, and they're gonna generate more metalloproteinases that they're gonna increase also the collagen breakdown. So all of these, they're gonna make a thinner fibrous cap on top of this necrotic core, which is gonna make more an unstable plaque. Also, because these smooth muscle cells during this progression of the atherosclerosis, they're gonna die, as well as the macrophages generated this big necrotic core together with macrophages foam cells. Next. So here is just a cartoon that I wanted to show you, because as we're going to talk about atherosclerosis lesion analysis, I wanted to show you how or where exactly where we are looking to the atherosclerosis lesion analysis. And here you have the aortic root, the entrance of the aorta to the heart. Then you have the ascending aorta, the aortic arch, and the descending thoracic aorta. And also to point it out, the brachiocephalic artery, where is also a place that you can also quantify atherosclerosis. Typically, this is the three leaflets that you're gonna find in the sections. Next. And to be more exact and more precise, here in this cartoon, you see the entrance of the aorta to the heart, and the section starts, if you want, to the beginning, the number 9, and you have correlations of the sections until number 1, where the plaque is almost close. And what we typically do is quantify different positions at the beginning, in the middle, and at the end, and you do a lesion quantification in different places. Next. So typically, for example, for the lesion area quantification, you can do hematoxylin eosin staining, and as you can see here in the first figure, you see the lesion in the dot black part, and you quantify this area, you measure in millimeters square, and you have this in different sections and different places. Also, you can quantify the lipid neutral content in a plaque. So you do all retom, the second images. And then another typical staining that you can do is the Mason trichrome, this third one that allows you to quantify the fibrous cap that you're going to find on top of the plaque formation. Also the necrotic part, which is the white part inside of the plaque. Then if you want to have the amount of macrophages in the plaque, you can do CD68 staining, or the F448, which is another marker. Or also, if you want to quantify the vascular smooth muscle cell, you can also do these stainings. Also, another typical one that we use is the picrus red. This red on top and on the bottom. You can quantify the collagen content of this plaque using the polarization microscope. And in the case that you want to check the expression of one specific microRNA, we have the fluorescent in situ hybridization, which is the FISH technology. And as you can see here, it's an example. We can find the miR30D expression in those plaques. Next. Also, we can also take the aorta, and with the aorta, you can do different things. For example, as I told you, you can quantify the plaque in the brachiocephalic artery, which is here in the first image. And typically, you can measure plaques in the aortic root and in the brachiocephalic artery. Also, you can keep the whole aorta in PFA and formalin and do an oil reto staining and on phase, where you can quantify the amount of neutral lipids, as you can see here in the bottom with these arteries. These aortas, sorry. Then you can also keep the whole aorta for gene expression and check the target genes, for example, as well RNA-seq. And in this case, you keep the aorta at -80. Or also you can do a flow cytometry experiment if you want to detect specifically, I don't know, if macrophages are expressed or less, or are in the whole aorta. You do different kind of digestion and different stainings, and you can get images like the one on the bottom and check the expression of one specific cell. Next. So as you know, microRNAs are the small single-strand non-coding RNAs. They regulate gene expression post-transcriptionally, and a single microRNA can target over 100 of RNA, and also it's implicated in health and disease states. Here, this is the biogenesis of these microRNAs that are translated in the nucleus thanks to the RNA pol 2, and then they go with different. They cut the whole thing in different steps by the drosha or the dicer. So at the end you get only one single strand. This single strand is gonna attach to the risk complex, and this risk complex together with a single strand microRNA is gonna bind because of pairing of the sequence, the seed sequence that it's typically 7-8 nucleotides that recognize an mRNA, the 3' UTR of an nRNA. So this risk complex and with this microRNA, there's gonna bind to this mRNA, and it's gonna degrade or inhibit the translation. Next. So this is also one cartoon that is almost the same as before about the biogenesis of microRNAs, but here I wanted to point it out that you have the option to increase the microRNA expression, and what you're gonna do is to use microRNA mimics. So, and with that, you're gonna have a decrease in the protein expression. Or on the other hand, if you want to decrease the expression of one microRNA, you can use the inhibitors or the antagomirs that they're gonna suppress the protein, sorry, they're gonna increase, so it's gonna suppress the microRNA. So for that reason, it's gonna result in an increase of the protein expression. Next. So how the microRNAs work? So it's like one microRNA can target different steps of the same pathway, or on the other hand, you can have different microRNAs that also interact or inhibit different steps of the same pathway, giving the importance of the potential of microRNAs regulating a biological process. Next. So, and with that, I'm gonna do an overview of microRNA research tools that we typically use for isolation and detection, for the target determination, for the regulation and the clinical application. Next. So the isolation is the typical isolation of mRNAs with a phenol-based extraction. So this is not novel, but what you can do for the detection is to use the microarrays that is going to give you the relative expression. So, and this allows you to measure the expression levels of a large numbers of microRNAs simultaneously. And these determine the relative change in expression between states, for example, treated versus not treated. And you can see how different is the amount of microRNAs. Here, one of the limitations is the sequence information. That means that you have to have this sequence information. So, and here you have the proofs that you have the sequence, and you're going to be able to determine if this microRNA is expressed or not. Then we have also the deep sequence. So this is the next generation sequencing that it's going to give you the absolute abundance, and it allows you to discover of new microRNAs. But one of the limiting steps is the generation of a large and complex data for library assembly, and also the cost, although now it's cheaper than when starts the deep sequencing, but this is one of the limiting steps. Next. So, of course, the detection of the real-time qPCR. So, for example, if you want to detect, in this case is the miR33 expression in the livers of these mice that they were under chow or high-fat diet or statin treatment, and you can do using the real-time qPCR the expression of this miR33. Also here you can have the relative expression of miR33 in different tissues. Then also this northern blot technology that allows you to have the information of the RNA detection, and here in the figure you are able also to determine the pre-microRNA and the mature form, in this case of 148, and also as a control you have the 5S of the ribosome. To do the northern blot, you're going to use radioactive probes. And also, and as I told you before, you have also the in situ hybridization, the fish, that you can detect the expression of miR21 in atherosclerotic plaques. Next. Then you have the computational approaches to microRNA target identification that, as you know, there are a number of different bioinformatic target prediction programs that will predict whether a microRNA can potentially bind to a target mRNA. And here you have 3 different ones, the Miranda, the target scan, and the Picture. This combination of these bioinformatic programs you can put together and have the information. In this case, it's 51 target mRNAs that, together with your own information of your microarray, for example, you're going to find, or you're going to end with 5 potential mRNA targets that, of course, you're going to need to validate through the UTR analysis, or the luciferase assay that I'm going to explain later how it works. Next. So this is only an example of the target scan that one of the bioinformatic programs that typically we use. And here you can select the species that you want to work, and also here you can enter the human the human gene symbol, or at the end the end of the page you can see the where I write the miR33, so you can enter your interest microRNA. Next. And then you're going to have the list of the transcripts that are related or potentially have this target of miR33 genes. So at the end, for example, I put the red thing to focus on the ABCA1, that you can see that it has 3 conserved sites, and which is also important because it's going to tell you that the more or less important can be this RNA. Next. On the other hand, if you can do the other way around, you can put the ABCA1 gene symbol. Next. And then you're going to have here the 3' UTR of this ABCA1, which has a this length, and appears the microRNAs that are supposed to be or potentially related to this gene. And you can see here how conserved is this microRNA miR33 in the ABC A1 3' UTR, which it tells you that it has potentially really important function since it is conserved among vertebrates. Next. Yeah, next. One more. Yeah. So one of the target determination that typically is used is the luciferase assay, and you're gonna have the 3' UTR of the gene that you want to study in the reported vector close to a renin luciferase and the promoter. And you're going to transfect this with a microRNA mimic that you are interested, or with a non-targeting control. And you're going to have a different light signal. And this is going to tell you how much this microRNA is bind to the 3' UTR of your gene of interest. Next. In another way to express, you can also have the target 3' UTR with a scramble, so it's not gonna bind, and you're gonna have this kind of a signal, the luciferase signal, because of the endogenous microRNA that is gonna bind to this target 3' UTR. But when you treat with a mimic, this mimic is gonna attach to the 3' UTR, and you're gonna have a less signal of luciferase. And another experiment that you can do, that when you treat this with inhibitors, they're gonna attach to the endogenous microRNA. This is not gonna bind to the 3' UTR, and you're gonna have an increase, much more luciferase signal. Next. And then also what you can do, or typically you do in these luciferase experiments, you do a point mutation of this 3' UTR. So the microRNA is not going to be able to attach, and this is going to tell so you're going to recover the signal, the luciferase levels, and it's going to tell you that there is really binding of this microRNA with this 3' UTR of your interest. Next. So another, apart from the target mRNA and the protein changes, you have also the luciferase reported assay that I told you. But apart from these, other technologies appears to be, or try to be, more specific. So we have the immunoprecipitation, which is the pull down of the risk components of this microRNA and mRNA union. To improve this immunoprecipitation, there is also the cross-linking and the immunoprecipitation, what we call the CLIP. You use the ultraviolet radiation to cross-link RNA to associated RNA binding proteins prior to do the typical pull down or immunoprecipitation. Then another improvement is the clip and the sequencing of hybrids, what we call clash. And this is the RNA protein complex that are UV cross-linked and affinity purified. Then these RNA hybrids are ligated, isolated, and reverse transcribed into cDNA. And then you deep sequence this cDNA, and it provides like a high resolution chimeric reads of these RNA and interactions. And finally, you have also the biotin streptavidin pull down, that it can specifically pull down a target of a single microRNA. So all of these techniques are want to improve that this union, that this interaction is real and there is no co-founders effects there. Next. Okay, then another thing that you can do if you want to check the microRNA function in vivo is to use the tissue-specific function. And to do that, you're gonna use conditional knockout mice, for example. If you want, for example, a knockout mice that does not express miR33 in the liver, you can have these mice and do your studies. So these specific conditional knockout mice are really interesting. And also what we did also is the target specific function using an mRNA target mutant mice. So this approach, what we use is the CRISPR-Cas genome editing to, in this case, for example, to selectively excise the region of the ABCA1 3' UTR containing these 3 bindings sites for miR33 that you can see here in the wild type. And you replace this with a construct in which this microRNA binding site had been modified and that prevent to bind. So with this approach, you really focus on these ABCA1 binding sites of the miR33 attached there. So it's another approach that you can also do. Next. Of course, then you can do the functional effects of microRNAs and their associated target genes in vivo or in vitro, using the mimics or the inhibitors, as I told you. So, for example, here in mice, you can inject with LNA, in this case, antagomir 148. With antagomir, you're going to block the expression of 148. And as you can see here in the B, there is a decrease or almost nothing expression of miR148. And also you can check the expression of a couple of targets of this miR148, which are ABCA1 and the receptor LDL. And you see in the LNA148 treated mice, you see an increase of the expression of these target genes. Also, in the in vitro studies, you can use the mimics. Here in the D panel, you see when you treat the cells with overexpressing this miR148, you see a decrease of the expression of the ABCA1, because you have more 148 that is going to attach to the ABCA1, and there's going to be a repression of the expression of this gene, one of its target. On the other hand, if you inhibit this microRNA, you're going to have an increase of expression of this ABCA1. Also, you can do functional assays. In this case, we perform a cholesterol efflux, and before the cholesterol efflux experiment, you transfected the cells with the miR148, so you are overexpressing this microRNA. If you are overexpressing this microRNA, it's gonna be attached to the ABCA1. ABCA1 is not gonna be able to do the efflux, and you're gonna see here a decrease of the efflux in the panel F. On the other hand, if you, before the cholesterol efflux, you inhibit 148, you're gonna have more expression of ABCA1, and therefore you have an increase of the cholesterol efflux, as you can see in the panel E. Next. So, and of course, there are clinical applications because microRNAs are very attractive therapeutic targets that can also be used as a biomarkers for certain disease. and you can have in the blood concentration a certain amount of microRNAs that you can really quantify, and they can, of course, serve as indicators of the pathology or certain physiology situation. And you can control the variations observed between patients or between treatments. So there is really an interest on this clinical application. Next. This is just show you a list of microRNAs and possible targets associated with heart disease that I'm not going to go through, but just to show you the data that is published in humans. Next. And also, of course, the potential application of microRNAs in the clinical practice. And here, this is a table with a major microRNA-based therapeutic which are in the development phase. And for example, you have the miravirsen, which treats the hepatitis C virus and targets miR-122. And this therapeutic molecule is already in phase 2 in clinical trials to check the safety and the safety. But on the other hand, for example, the MRX34 is also a therapeutic molecule that was in phase 1 clinical trial that target miR34A, and it was used for treatment of different types of cancers. But this clinical trial had to stop because of the side effects, and they didn't go through phase 2. So, and again, here you have most of them are in preclinical stages. So there is still hope and room of to really see the potential application of these microRNAs in the clinical practice. Next. So this is just it is known that the aberrant amount of microRNAs has been linked to the formation or related to the formation of the plaque. And you can see here how can affect different cell types that I've been told before that are important for the atherosclerotic plaque formation. And here you can see how can affect the function of the endothelial cells, as well as the migration or the proliferation of vascular smooth muscle cell, as well as also modify and it's important for the function of macrophages and the foam cell formation. Next. So, and now I'm going to talk about the one microRNA that is the miR33, which I've been working for a while this in my last period of time in the States. And this microRNA, it's a microRNA family that consists of miR33A and miR33B, which it is well known or it is well characterized and used as an example of microRNA that is mediated the regulation of lipid metabolism. And miR33A and miR33B are intronic microRNAs that are co-transcribed with its gene SRBP2 and SRBP1. And exactly for example, the miR33A, like other intronic microRNAs, it's co-transcribed with his host gene, which is SRBP2, and with this co-transcription, miR33A, it's gonna block the expression of genes related, as I told you, to cholesterol efflux and the HDL biogenesis. And this makes that the cell, or protect the cell that is already with low levels of cholesterol, to a possible loss of more cholesterol. So it's like a double check or double protection of the cells, right? Because of course SRBP2 is gonna target other genes like MNG co-reductase that is related with cholesterol biosynthesis, and also the uptake of lipoprotein with the LDL receptor. But on top of that, you have also the miR33. Apart from controlling the homeostasis of cholesterol, miR33 is also important controlling the fatty acid metabolism. As you can see here, SRBP1 is co-transcribed in the miR33B, and this is going to target different genes that are related with fatty acid oxidation. But apart from that, next, we have also other critical processes that are regulated by miR33, which are the cell cycle, the insulin signaling, the inflammation, autophagy, mitochondrial, liver, and kidney function, that some of them are also related with atherosclerosis. So miR33, as you can see, is really an important microRNA. Next. And here it's a summary of how miR33 regulates the lipid metabolism. As I told you, in the macrophage, miR33 is blocking the ABCA1 and G1, so the cholesterol efflux. Also, in the liver, miR33 is blocking the HDL biogenesis through the ABCA1. Also, it's blocking the fatty acid oxidation and also the insulin signal, and also plays an important role in the biliary excretion. So here you can see that miR33 represents that you're going to have less HDL biogenesis, less cholesterol efflux, and less bile acid secretion, which is going to converge and having less reverse cholesterol transport. Also, miR33 is going to have less fatty acid oxidation, which is going to bring you like a more VLDL production, which is going to increase the triglycerides. And also, miR33 is regulating insulin signaling, and that's going to give you some insulin resistance. Next. So what a lot of clinical trials try to increase, to raise the levels of HDL, or they really did that in different terms. So you can use the lipid-free ApoA1 infusion, or the HDL infusion, also the decrease of the catabolism, or also the using of CTP inhibitors. But the problem of they were able to increase these levels of cholesterol, but they did not appear to have an impact on the clinical outcomes in patients with cardiovascular disease. So what we think next is that if miR33 is controlling the biogenesis next, and ABCA1 and G1, what we can do is inhibit this miR33. So yeah, next. So what we did, different groups, we used this antagonist of miR33, and we were able to increase circulating cholesterol HDL using different approaches like lentivirus or LNA oligonucleotides or adenovirus, and also in different mice, in different animal models, for example, mice or non-human primates. More important, using this antagonist of miR33, we were able to attenuate the progression of atherosclerosis and also enhance the regression of this plaque. Next. What we did, first of all, is use the global genetic deficiency mice of miR33. And interestingly, what we got is some pro-atherogenic factors. So we had an increase of insulin resistance, increase of cholesterol, VLDL and LDL, and increase of the body weight, and no changes in the atherogenesis. But then we decided to really use the hematopoietic deficiency miR33. So, and we found that these anti-atherogenic factors, such as increase the cholesterol efflux, increase the reverse cholesterol transport, less lipid accumulation, more atherosclerosis, and less inflammation. And more important is that we saw is the less plaque, atherosclerotic plaque formation in those mice, telling us that the importance of the specific inhibition of the cell type or location, or also in time-wise. So it's important to be aware that if you do a global knockout, maybe you're not gonna be able to see how is working in another type cell that can be more important than the global. So this is the thing that you have to be aware which cell or which region you want to, in terms of therapeutics that I'm going to explain, you want to direct your anti-miR or your overexpression of one microRNA. Next. So for that, we have some different microRNA-based therapy in atherosclerosis that it has been used. And for example, we have the viral vectors that you can have adeno-associated virus or retrovirus or lentivirus. And it has the advantage that there are stable transgene expression, and it has a high efficiency. But the disadvantages are that the high carcinogenic potential and the excessive immunogenicity. Next. Here we have one example of the use of these viral vectors as a microRNA-based therapy, that they use the antagomer 33 containing endothelial cells derived exosomes that enter neighboring intimal cells. And in this case, they use the helper adenovirus. Then we have also the exosome carriers, which are extracellular vesicles originating from the endosomes. And it has the advantage that it is high stable in circulation and is not immunogenic, although the problem is that the difficulties to isolation of the exosome and the high cost to do that. Also, we have the nanoparticles. We have you can have polymeric nanoparticles or liposomes, and here the good thing is that are easy to manufacture and it's high packaging capacity, so you can put a lot of things inside. But the disadvantages is the cytotoxicity and the low delivery efficiency. And here we have a couple of papers that they use in the first place the chitosan nanoparticles together with different microRNAs, and also the liposomes that encapsulate miR-146 that they show that they reduce the vascular inflammatory response, but this was made only in vitro studies. Next. And also you have recombinant HDL as a nanoparticle, and which actually I'm working with that as well, because I would like to see, indeed, we use this recombinant HDL together with anti-miR therapies, and we are doing the in vivo experiments. So next time, maybe I can tell you more about that. Then we have the polyelectrolytes complex micelles that I that has a lot of stability and allows you to the drug release in a controlled manner, but it is complex to characterize these cells, and it takes a lot of time to process, to get these cells. And finally, we have this pH low insertion peptide, which has this selective delivery in acidic conditions, and it's high bioavailable. But as a disadvantage is the accumulation in other tissues and requires further optimization. And next, and this I'm going to tell you more about this peptide that we use to attach the anti-miR33 therapy. Next. This peptide is the pH low insertion peptides, which are a family of soluble approximately 36 amino acid peptides, which it binds to the membrane surface. And you can see here in the regular state in the B panel or state A, it's gonna stay there in the membrane. But when there is a drop of the pH, it forms an inducible transmembrane alpha helix under these acidic conditions. And then it has the ability to translocate the membrane impermeable molecules into the cells via a non-endocytic route, which is important. Next. And this peptide was first used in a cancer model, and they attached this peptide with a fluorochrome, and they injected to two different models of cancer. And as you can see here in the panel A, you have the control with no signal at all, and the peptide, you have a lot of signal in the tumor, which, as you know, is an acidic microenvironment. No signal in the liver, which is good, and some signal in the kidney, because, as you might know, kidney is also an acidic tissue. And here the image of the mice where the tumor is located, you have the big signal of the peptide attached with the fluorochrome. In the B panel is another kind of mouse model with a lymphoma, and again, where the tumor is, you have the more signal. Next. Next. So what we thought is that atherosclerotic plaques are supposed to be an acidic tissue microenvironment, and we found different bibliography that goes in that direction. It has been said that established atherosclerotic plaques have a low pH environment, that the epoxy occurs within the atherosclerotic plaques, and also that macrophages convert most of their glucose into lactate. So next, knowing this, what we prove is that the delivery of this peptide conjugated with fluorochrome into an atherosclerotic plaques. So we put LDL receptor knockout mice 12 weeks on Western diet. So we generate a plaque in the aorta and the aortic root. Then we injected the control or the peptide. And different time points we take blood, and also we sacrifice some mice at 4 hours, 12 or 24 hours of the injection, and do the ex vivo imaging. We did the intrajugular injection, but then I'm gonna show you other kind of injections, because intrajugular, of course, goes directly to the circulation, but it's gonna be complicated if you want to do, or if you think to do a therapeutic treatment. But the first try we did with intrajugular injection. Next. So this is the jugular injection of the peptide with the control and the peptide attached with a fluorochrome and do the ex vivo imaging. And here you can see in the insert the aorta and the heart, and you can see some white spots, which is where the plaque is located. And as you can see in the peptide region, the peptide mice, you can see the red signal telling that the peptide was going to where the plaque was located. In the control, that you can see no signal, but you see the plaque there, this white thing. Also, some signal in the kidney with the mice that was treated with a peptide, and not too much in the liver and the other organs. Next. So we did also this time course, and as you can see, 4 hours is where we have more signal, and at 12 and 24 hours the signal is decreasing. Next. What I tell you before, we did also retroorbital injections in comparison to the jugular, because if you think to do a treatment, intrajugular is going to be complicated and you're going to be only able to do it twice. Retroorbital, you can do more injections. So, and as you can see here, we have signal both in the retroorbital as well as in the jugular or intravenous in comparison to control mice. Next. So then in another set of experiment, we also inject with a peptide, in this case with this fluorochrome, different from the other one, but just because we wanted to take the tissues and do sections of these different tissues. So here we have the aortic root sections, and in the left you can see the mice injected with a peptide. You can see this red signal of the peptide that is kind of colocalizing with the CD68, the green marker of macrophages. And in the control, we don't have any red signal. Next. Also, in the same experiment, we took the liver, and as you can see here in the control, we have an amount of signal. This red thing is the same as the ex vivo imaging, that you have more green and some yellow there, because of the control peptide, which has protons. So, but then in the liver of the peptide with the fluorochrome in the liver, we cannot see as much signal as in the control, although we have some signal there. Next. Also, we take the kidneys, and as you can see in the ex vivo imaging, the kidney has quite of a signal, as well as in the section of the kidney. Next. So, and also another experiments that we did, we took more mice, and we inject more mice with on diet with the plaque already formed in the aorta and the heart, and we inject this peptide, and we took the whole aorta. We did the digestions and the staining for the macrophages to see if this cell type was able to really get the peptide. And as you can see here in the bottom, the green signal is the peptide where macrophages were getting this peptide. We only checked the macrophages. We didn't do the experiment using checking how endothelial cells or vascular smooth muscle cells were able or not to get the peptide, which is something that we should do. Next. And this work was, this peptide was used in a mice model, and we were able to suppress the miR33 using this peptide, and we saw that there were an improvement of the atherosclerotic regression in this case. Next. And also since we saw that the peptide was also getting to the kidney because it's an acidic microenvironment, we use this technique, this peptide, to inhibit miR33 in the kidney as an example of another disease that we were studying. Next. Yeah, next. Okay, and just to point out go back one, please. Pointed out some aspects and limitation of miRNAs that I think it's important to say that some of the or many human studies that have limited patient numbers, so it's difficult to have a conclusion there. Also, it's important the conditions for obtaining miRNAs and their detection can be different in different studies. So this also it's important to have to keep in mind. As I told you, one microRNA can regulate hundreds of genes, and one gene can be regulated by many microRNAs. So we have to be aware of target effects, and this is the importance of to know, or then if you want to focus in one cell or inhibit one specific microRNA in macrophages or endothelial cells. So these things that you have to be aware. Also, the possible deleterious effects on other tissue depending on the administration methods. And of course, the stability of microRNAs and their release, you have to be aware. And next. And with that, I would like to thank the different institutions that I've been working, the people that help me in these projects, and the fundings. And with that, I'm gonna take questions, any doubts or...
Wow, thank you so much for this elaborative, I would say, lecture on all the types of manipulation we can use for microRNAs as targets, as markers, and so on. I have so many questions, but I always like to give others opportunity to ask. So we can start with Fabio. Fabio, I will try to unmute you, and then you can ask your question directly, so we don't bother with chat.
Can you hear me?
Yes.
Okay, so thank you, Naomi. Really beautiful work. So my question is, I'm really intrigued about this pH lip technology. So wondering whether this can be used to target ischemic tissues. I don't know, for instance, if atherosclerosis goes bad, then you have myocardial infarction. And here I can see on the one side that the more acidic part of the tissue will be also less vascularized, so there may be an accessibility issue. So have you tried to do any of these kind of experiments, and where is the balance? So because theoretically, if you can target the border zone, for instance, with something that is pro-angiogenic, could be really an element of specificity that could be very important.
I didn't try in other different models or the ischemic model. I'm thinking in the fibrosis kidney disease example. So I'm not an expert in the kidney, but I guess that it's also, you know, you get a fibrosis and you don't have too much vascularity there. So, and it was working, I was going there. But again, I'm not sure about you know the specific one that you told me about the ischemic tissues, or I mean, you can try. I don't know. No, I'm not sure. I'm not sure.
All right, thank you.
Welcome.
May I ask, I have a comment and a general question. First of all, clearly this was an illuminating presentation for us, for me, because I mean, there are so many angles that you can see, from which you can see the system. And so this made this many opportunities, but of course, a lot of, obstacles maybe. General consideration is that, of course, each animal, specific animal model is developing a specific type of plaque. This means that if we do a kind of translation, quote unquote, if it is possible, from one animal model to humans, we should you should classify and select the specific type of plaque or targets, or let's say cell type target, that is somehow the best target also in humans. Let's say we want to target endothelial cells, we want to target their dysfunction, their adhesion molecules, whatever. So if this is something, I don't think this is too much in terms of translation, but in order to get the maximum efficacy, you should kind of classify the type of plaques. Also, we know very well, all of us, that, probably we cannot have in animals, the plaques that develop over decades in humans with a very thick fibrous cap and so on. So in a way, it would be nice to have a sort of, parallel, let's say, in order to do not just treat humans whatever they have, but to treat a subset of humans with specific homogeneous types of plaques in order to get a reasonable efficacy.
Yeah, but I think that this is going to be complicated. It's not so in mice. It's it's more easy to work with different cell types and focus and see what's happening. In humans, I don't know. In the clinical trials that they have, it's more the middle 122. It's more in the liver, or it's more located. So if you have one microRNA that is important, that targets one specific gene in one organ, or this is going to be easier. But and again, the atherosclerotic plaque in mice and humans, which I didn't say, it's different and they behave different. So I think that we are still kind of far away to get to the humans, but I mean, this is why we are doing research, right?
The other comment, the other question is, you showed different techniques for target determination. And of course, each of them, there are pros and cons and different cost probably also. Can you make a comment? I don't say a little suggestion, just, late afternoon or in the evening, because we are like kind of late, but a comment on that.
On that.
Pros and cons. What you showed, for example, from UV cross-linking to, let's say, biotins that targeting, I mean, so.
My experience, we always did luciferase assay. That for me was really nice because it's like interaction, microRNA, the 3' UTR of your gene. You have more or less signal. This means that it's attached or not attached.
Yeah.
But lately, really, people are focusing more in the pull down, really, the unions that are happening there. So I guess that I have to move to that technology. So I've never used it before. So I cannot tell you. I know that it's not easy. It takes so the protocols I remember, like long protocols. Not remember if they're expensive or not. But I didn't go to these technologies, so I cannot tell you more about them because I'm more in the luciferase assay, which for me it's working. It's kind of easy and yeah, but I guess that I have to move also to the pull down or the immunoprecipitation, the PAR clip, and all this. But I have no experience, so.
Thanks. Very interesting. Thank you so much.
I should move to another lab that they're doing that and learn. So yeah.
Very nice. Thank you so much, really.
Dimitris, I guess you have also many questions.
Yes, I have one question. Hi, Noemi. Excellent presentation. Thank you very much. I have a question about the methods of delivery that you mentioned. One of the methods you mentioned the reconstituted HDL. And I have a question about this. First of all, what are the advantages of using reconstituted HDL compared to the other methods that you mentioned, especially the last one? What are you targeting with reconstituted HDL? And do we know how the miR will insert it into the cell? Because we know that HDL is not endocytosed. I mean, it just takes cholesterol and goes to the liver and delivers cholesterol and then is remodeled. Do we know how the miR goes into the target cell from a reconstituted HDL, or is it conjugated?
The reason why we start doing that, we thought because HDL is, these anti-atherogenic properties that has itself, and it was like, okay, these anti-atherogenic properties, this HDL goes also to the plaque. Why don't we use this? Also because you can get a lot of this recombinant HDL. Is not expensive, so we thought, kind of an improvement, and use this recombinant HDL with anti-miR therapies. Also, if we can avoid the off-target effects, or if it can go directly to the plaque, as HDL goes there and remove the excess of lipids. But I have to say that we are not sure if, so in vitro experiments, I incubate this recombinant HDL with the anti-miRs, and they are blocking the expression of ABCA1 and LDL receptor, because I'm trying with a 148 microRNA. So in vitro is working. The cholesterol efflux also is functional. So it looks like the recombinant HDL is working, but now I'm doing the in vivo. So we're going to see if we're going to be able to see less plaque formation. Also, I would like to do some cytometry experiments to see if these macrophages are taking up this recombinant HDL. So we are on the beginning of this project. But yeah, we wanted to also, because the pH lead. So I mean, it was a collaboration with some people in Rhode Island. So now here, I'm in Spain, and I was like, okay, let's try another thing, and we focus maybe more in the recombinant HDL, but still in the beginning of this project.
Thank you. Thank you.
You're welcome.
Thank you so much once more for really discussing all the aspects that are currently, I think, very burning subjects, because we know that there are some therapies, and also Thomas Stum Lab, they developed I think, miRNA mimic for heart failure, or some version of heart failure, and they are now acquired by Novo Nordisk, I think. So obviously the technology is advancing. And as you mentioned in the early stage of your presentation, microRNAs are not specific, so they're relatively easy to target from the theoretical perspective. But the key thing might be in the delivery strategies, how to really target specific tissues and to find a version. And I think that I'm really looking forward to hear your experience with recombinant HDL, to see how this works, and can it really bring it to macrophages and to plaques. I will focus a little bit more on more basic questions, because this is something that we found in literature so many times, and people that are new in this field are quite confused sometimes with this. And since this is more or less aiming to kind of PhD students and early postdocs and people who are just interested in this field, there is this a confusion with the names of microRNAs. Sometimes you can see microRNA XY, like 33, and then you can see 33A, 33B, and then you can see 33P, 5P, and so on. So maybe just if we can elaborate a little bit on this nomenclature, and also because we know that microRNAs are synthesized as in duplexes, and then they are cut in the final stage, and then you get 3P or 5P version of microRNA. Do they act similarly, antagonistically? If you just can emphasize a little bit about this part.
Yeah. So so the microRNAs, you can have what we call the microRNA family. So you can have the whole thing, and then different sequence that can vary only one nucleotide, and then you can have and they're different, and their function also can be different. So you really need to be whenever you're going to do one experiment or a sequence or something, you really need to be sure about the sequence of your microRNA of interest. If not, you're going to look for other things. Then you have at the beginning is a double strand, and then with Drosh and the dicer, you can have only one strand, but the other one, what we call the, how do you call it? Well, the passenger strand is different from the mature form, but also plays a role, and you have to really check that microRNA that can act differently from the mature form. So you have to be aware that your microRNA, which with, for example, the bioinformatic things, whenever you want to look, you want to have the information of the different 5P, 3P, and you have to really know which one are you looking for, because the sequence is not going to be identical and can have, of course, different functions.
And regarding the part of the mRNA where the microRNAs are targeting, we know it's mostly 3' UTR. It can be though 5' UTR. It can be in the middle, in the coding region. So are there any kind of conclusions, which microRNAs are more prone to act on 3, and what in the functional sense will be the result of acting on 3' UTR or 5' UTR, or still it is quite elusive, what is the difference? And
I don't know. I have to tell you that I'm not sure.
Because I was quite intrigued that you were focusing with the CRISPR thing on the 5' UTR, which is still something that is quite often.
Yeah, well, in this case, because in the ABCA1 3' UTR, the 3 in the 5' is where you have 3 seed regions that where the miR33 is going to touch. So we thought, okay, because it's really long, you cannot have the whole thing. So we focus on those that you have the 3 places where miR33 is attaching. Of course, and we have this question on the reviewer, what about the other one? But then, well, it's also, it's another binding site and can do other things, but we focus in this region because you have 3 different binding sites of the miR33. That's the only thing that I can say about that.
And when we discuss the 3' UTR, they can vary very. The variation of the length in 3' can be quite fast between different genes and between different isoforms of mRNAs. So do you see any kind of correlation or association between the level of mRNAs that can bind to certain gene depending on the length of 3' UTR? For example, if the 3' UTR is longer, does it create more sites to bind from different microRNAs, or it's not that physiologically relevant?
I have no idea. I didn't check that. Now that you're telling me, it's like I'm from time to time you start looking to the target gene and you see the sequence, but I didn't pay attention on that. So this doesn't mean that it has a pattern or something, but I really don't know.
Okay. And then just like a future perspective question from my side, which type of delivery systems you think it's most promising when it comes to microRNAs and atherosclerosis? We know that many of these delivery systems for RNAs are mostly focusing on liver, and it's relatively easy to target liver because liver just absorbs all these types of particles. But if we want to go more on atherosclerotic plaques and maybe, you know, target those things, do you have any favorite approaches, or you are just everything is an open book and it's just
you want one that is working, and in the case, for example, of miR33, you really don't want to go to the liver. So because you have an increase of triglycerides or the VLDL, so really you don't want to have the delivery there. So yeah, anything that goes to the plaque, it's going to be fine.
Perfect. Okay, so that's enough for me. I don't know if we have more questions. I don't see anything in the.
I only had a comment from because this is today we celebrated the 10th webinar with Noemi.
Yeah.
So I personally, on behalf of all of us, thank you for organizing the series.
Yes, thank you for.
So Miran, thank you very much.
No problem. It is my really my joy to witness all of these webinars and really discuss all these things, because it's something that we cannot really, you know, there is so many things and so many papers on different topics and different things, and it is really nice to have and true experts in the field that they can really summarize all these concepts and really present us in a more like a coherent and, you know, structured way, so we are not completely experts in the field, we can follow a little bit more and get a lot of information out. So for me, these 10 webinars were really joyful things to organize, and hopefully we will have more to come. Now I'm just checking when is the next webinar. It's going to be on the 25th of June. And the title is going to be a little bit different, so we are kind of diverging from RNA topics for a while. It's going to be which omic techniques to choose for biobanking samples, and it's going to be led by Florence Pinet. And then we will make in July and in August we will make a break to get a little bit of rest for all of us, and then from September probably we will continue with the webinar series. So if there are no other questions, I would like to thank you, Noemi, and everybody who was here today, and see you in basically 1 month for those who are coming to atherosclerosis Congress. See you there.
See you there.
Looking forward to continue with discussions there.
Yeah. Thank you very much. Bye.
Have a nice week, everybody. Bye-bye.
Bye-bye.